our objectives for today is to distinguish between observational studies and experiments. What's the difference? We're going to talk about the language of experiments and how we set them up and really how they work. The three principles of experimental design, that'll come later, as well as randomized block design and match pairs design. We'll hopefully touch on completely randomized designs today. Observational study versus experiment. An observational study does this. It observes individuals, and measures variables observes individuals and measures variables of interest. I need a ladder. But does not attempt to influence the response. It says you observe individuals and you measure variables of interest, but you do not attempt to influence the responses. Experiment, on the other hand, this one's shorter. What's an experiment do? It deliberately imposes a treatment. Deliberately imposes some treatment on individuals to measure a response. Deliberately imposes some treatment on individuals to measure a response. Observational study, we're just like watching and measuring some results. With an experiment, we're doing something to people. We're saying, you 10 people are in this group and you get caffeine in your pop. You 10 people are in this group, you have pop but no caffeine in it, and then we measure the responses. So we're specifically putting in people in groups. With an observational study, we're just like kind of observing what happens. Grab your books and turn to page 233. And I should make note of this before we dive into the example in the book here. It says here, when our goal is to understand cause and effect, bless you, experiments are the only source of fully convincing data. So if you want to talk about cause and effect, the only way you can talk about cause and effect is with an experiment. Look at Check Your Understanding, page 233. Number 1 says, does reducing screen brightness increase battery life in laptop computers? To find out, researchers obtained 30 new laptops of the same brand. They chose 15 of the computers at random and adjusted their screens to the brightest setting. The other 15 laptops were left at the default setting, moderate brightness. Researchers then measured how long each machine's battery lasted. Was this an observational study or an experiment? And justify your answer. Experiment, because you are imposing a treatment, right? Some of them you put in this group and you change the brightness. Others of them are in this group and you just kind of see what happens. Questions 2 through 4 refer to the following setting. Does eating dinner with their families improve students' academic performance? According to the ABC News article, teenagers who eat with their families at least 5 times a week are more likely to get better grades in school. This finding was based on a sample survey conducted by researchers at Columbia University. Observational study or experiment? Observational study, because you're just observing what happens. You're not assigning people to any groups to do anything in particular. Number 3, explanatory and response variable. Keep your book open because we're going to come back to this in a second. An explanatory variable may help explain or influence changes in a response variable. What's a response variable? It measures the outcome of a study. I'm want to take a step back even from that text and from these two for a second. If you have explanatory and response variable, the most simple, basic example that I like to give you is this. Depending how much it rains is going to determine how much the grass grows, right? So the more it rains, the more the grass is going to grow. Can we agree on that? Good. So in this example, this short, sweet example, my explanatory variable is how much it rains, and the response is how much the grass grows. What's going on? The amount of rain is going to explain your response, how much the grass is growing. Often, this is chapter 3 stuff. We skipped chapter 3, so I'm just throwing these two terms in here right now. It's going to be seen on a graph. It's like your explanatory is on your x-axis, and your response will be on the y. So when I look at this example in the textbook, when we were talking about those families and teenagers who are eating with them and their grades, what are we talking about? What are we trying to measure for your response? What do we want to know about them? Their grades, right? So your response, you're like looking at their grades and what's explaining their grades based on this study. Whether or not they eat with their family, right? So your explanatory, do these kids eat with their families? And the response, what's that impact, or how does that impact their grades? Number 4, explain clearly why such a study cannot establish cause and effect relationship. Why can't we establish cause and effect? Because it's an observational study, it's not an experiment. Plain and simple. You don't need to say anything more than that. Working variables, we'll talk about those in a minute. So observational studies often have something going on as well, even sometimes in experiments, talking about confounding and working variables. Have you heard these terms before? Some of you in AP Psych maybe? Confounding is the process of what happens when they're working variables. Here's a working variable. Here's the definition of working variable. A variable that's not among the explanatory or response variables, but may influence the response. So when I say how much it rains is going to determine how much the grass grows, what other predictors could be in there? Now, how much sunlight there is, temperature, fertilizer, right? Those are all called working variables. I didn't tell you about them before, but they're also playing a role. So you got fertilizer, you got the amount of sun, the soil, somebody else said something else. But those are working variables. And when you have working variables, we call that confounding. Confounding occurs when two variables are associated in such a way that their effects cannot be distinguished from each other. We don't know, yes, it rained a lot, but how much the grass grows, is that due to the fertilizer? Is it due to the sunlight? Is it due to the type of soil? Those are all called working variables, which results in confounding. See the big picture? Kind of, sort of? We're building there. Language of experiments, I know you guys have heard these before. An experiment is a statistical study in which we actually do something. When we're doing something, we call this a treatment. So in an experiment, we do something, we apply a treatment to people, animals, or objects, and when we do something to people, animal, or objects, we call these our experimental units. To observe the response. So that's kind of the summary. That's basically what it is right there. What's a treatment? It's a specific condition applied to the individuals. What are the experimental units? It's your smallest collection of individuals to which we apply the treatments. And we'll go through some examples in a second. And here, our human beings are called subjects. So if you have animals, well, like, they're experimental units. If we have plants, they're experimental units. If we're talking specifically about people, we call our people subjects. Just kind of differentiate there. And every now and then you're going to hear this. You're going to hear explanatory variables are referred to as factors. Not often, I don't talk like that. I just refer to them as explanatory variables. But they could be called factors, just so that you're aware. In an experiment, each treatment is formed by combining a specific value, often called the level of each of these factors. Again, it may come up every now and then, but your levels, I'm not going to go too in depth with that. Your first example, bottom of the page there. A study published in the New England Journal of Medicine compared two medicines to treat head lice. This question just grosses me out, but an oral medicine called ivermectin, and there should be the word "and" in there, and a topical lotion containing malathion. Researchers studied 812 people in 376 households in 7 areas around the world. Of the 185 randomly assigned to ivermectin, 171 were free from head lice after 2 weeks, compared to only 151 of the 191 households randomly assigned to malathion. Identify the experimental units, explanatory and response variables, and the treatments in this experiment. So, first of all, what are my experimental units? What are we doing this to? We have people, but how are we selecting those people? We're choosing those specific households, right? Because what's it say here? There were 191 households that were randomly assigned. So my experimental units are those households. We have explanatory variables and response variables. I usually like to start with like the response. So what are we trying to measure? What do we want to know about these households? Yeah, how quickly the lice goes away. So the response variable, what do we want to know? Are these households lice-free? So like the response variable, are these people lice-free? Are these households lice-free? And what are we doing? What's going to determine that? We're giving these different households medicine and like a lotion, right? We've got an oral medicine and a lotion form of the medicine. So if we just say the medicines we give them is going to determine if they're lice-free, you've got your explanatory and you've got your response. And then we get a little bit more specific, you could say, and we talk about treatments. What are the treatments that we give these two households, or these randomly assigned households? And probably nobody wants to say them because they're hard words to pronounce. Yeah, ivermectin and malathion. How about this next example? Next page of your packet. Growing tomatoes. Does adding fertilizer affect the productivity of tomato plants? How about the amount of water given to the plants? To answer these questions, a gardener plants 24 similar tomato plants in identical pots in his greenhouse. He will add fertilizer to the soil in half the pots. Also, he will water 8 of the plants with 0.5 gallons of water per day, 8 of the plants with 1 gallon of water per day, and the remaining 8 plants with 1.5 gallons of water per day. At the end of 3 months, he will record the total weight of tomatoes produced on each plant. Identify explanatory and response variables, your units, and your treatments. What are experimental units? Yeah, the tomatoes are your tomato plants. I guess specifically it's the tomato plants that we're watering and trying to get to grow. What do we want to measure? What's our response variable? The weight of the tomatoes. And what's the different things going on that's going to determine those weights? How much water you give them and how much, whether they get fertilizer or not. Yep. So the water and the fertilizer. So in this case, there's 2 explanatory variables that we're talking about. And lastly, our treatments. Here's where, for me, to kind of like draw a picture and figure out what's going on, it helps me kind of visualize it and process through. So first of all, he said that in half the pots he gives fertilizer, and the other half of the pots he doesn't give any. So I kind of draw a diagram. I'm like, these plants have fertilizer, these plants have no fertilizer. But then what else do we know? There's different amounts of water that each of those plants get, right? 8 of the plants get half a gallon, 8 of the plants get a gallon, and 8 of the plants get 1.5 gallon. So of those that get fertilizer, we're giving half a gallon of water. And those that have no fertilizer, they also have half a gallon of water. Right? 8 of the plants get half a gallon of water, but 4 of them have fertilizer, 4 of them have no fertilizer. So you might say half a gallon water with fertilizer, half a gallon water with none. Then we have 1 gallon of water with fertilizer, and we have 1 gallon of water with no fertilizer. And we also have 1.5 gallons of water we give the plants with fertilizer and without. So really, how many different treatments are there? There's 6 different treatments. So the treatments is where you kind of like specifically detail out how much of each of these plants get so much water and fertilizer. Here, your explanatory and your response are still the water and the fertilizer. It's just not as specific in a sense. It's not the names of your medicine. It's not the specific amounts those plants get. Let's keep going. How to experiment badly. So we're going to talk about how to experiment badly, but first there's 3 things that we really need to know about experiments. Experiments are the preferred method for examining the effect of one variable on another. So in other words, to see if there's a relationship between 2 things, to determine cause and effect, we use experiments. By imposing a specific treatment of interest and controlling other influences, so if we can control for those lurking variables as much as possible, we're going to better be able to determine what's causing what in this experiment. And lastly, good designs are essential for effective experiments just as they are for sampling. So in sampling, we talked voluntary response and convenient samples give you a lot of bias, right? It's bad, but it's a way to do it. And this first one here is an example of what's not the most effective way to experiment, but you can do it, right? Here we have a high school regularly offers a review course to prepare students for the SAT. This year, budget cuts will allow the school to offer only an online version of the course. Over the past 10 years, the average SAT score of students in the class was 1620. The online group gets an average score of 1780. That's roughly 10% higher than the long-time average for those who took the classroom review course. Is the course more effective? So what do? We took students, put them in an online class, and we measured their SAT scores. Is that effective? Is it more effective than the other way of doing it in the past? We had a handful of kids who were like, hey, I'll take an online course and do whatever. Is that going to be a lot more effective than in the past? What do you know about an online course? It's boring. It's very self-paced, right? You need to be very self-motivated to take a course like that, right? So when I look at this, and I'm looking at this online group getting a score of 1780, I'm assuming that that specific group of people, they're very self-motivated learners, so of course their score is going to be a lot higher. We don't have anything to compare this group to. You've got one group in the online class that's doing really well, but what do we compare them to this year? We don't have anything to compare them to, right? So in this one, you could say, like, there's no control. We don't know who to compare it to. And you also might say these students are very self-motivated. So this is the very most basic setup of an experiment. If it's in a lab with like lab mice, like lab mice, it'd be okay to do something like this because we have experimental units. And what do we do to them? We give them a treatment. And we measure the response. In a lab, there's so much control going on. There's so much you can control about temperature and food and everything for like a group of lab mice. So in that sense, it'd be okay. But when we get out and we start working with people or animals and you're actually out in the classroom, out in the field, out in different workplaces, and you're starting to interview and survey and experiment on people there, the conditions are so much more variable that your results are going to be different, and we can't just use something so basic like this. So outside the lab, badly designed experiments often yield worthless results because of confounding. You've got so many different things going on. I mean, just as far as maybe time that people eat, how much people eat, temperature in a day, if you're like looking at weight loss or something like that. There's so much confounding that we need to have a little more specific experiment when we go through and do this. I'm going to give you a minute. Read through that example on your own. Does caffeine affect pulse rate? Suppose you have a class of 30 students who volunteer to be subjects in the caffeine experiment that we just looked at. Explain how you would randomly assign 15 students to each of the two treatments. You have a group of 30 students. How are you going to say, you people in this group, you people in that group? Give everybody a cut up 15 slips of paper, put them in a hat, and have people draw, right? Won't that be simple, rather than using like a table of random digits? So you can maybe say, let's write A, which is like the caffeine group, on 15 slips of paper, and B, which would be your no caffeine, on 15 slips. Have students draw from a hat. That's probably the most simple, most practical way to divide people into groups. Here we go. How to experiment well. So a basic experiment, you get your experimental units, you give them a treatment, and you measure the response. In a lab, it's okay. The best thing you can do is to do a comparative experiment, and this is to help with confounding, with everything that's going on. So comparison alone isn't enough. What we want to do is we want to randomly assign people to which group they're going to be in. That's going to help you eliminate bias. The basic setup right here. What do? We take our experimental units. We randomly assign them, so random assignment occurs. You got your people. We want to randomly assign them into 2 groups, or 3 groups, or however many groups you have. So that gives us group 1 and group 2. You apply your treatments. And last but not least, we compare results. Sometimes it might not be 2 medicines that you're comparing. Sometimes you might have a control group. Have you heard of a control group? It's like your baseline. You're going to use that to compare to what you're doing. Work with me through this and then we'll wrap up. A health organization wants to know if a low-carb or low-fat diet is more effective for long-term weight loss. The organization decides to conduct an experiment to compare these 2 diet plans with a control group that is provided with brochure about healthy eating. 90 volunteers agree to participate in the study for 1 year. Outline a completely randomized design for this experiment and write a few sentences about how you would implement it. So this right here is an example of our experimental design. In our study, It doesn't, does it tell us how many people we have? Yeah, it tells us we have 90 volunteers. So we're basically going to fill in the blanks with the information that we're given here. My experimental units are so you have 90 volunteers. We randomly assign them to how many groups? What are we trying to get at? We want to know the varying diet plans. We want to know if a low carb or low fat diet is more effective, and what do we got to compare that to? Ideally, a control group, yes, we can compare the results. So I want to divide it into 3 groups. So you have group 1 is going to have 30, we're going to call them subjects because it's people that we're talking about. Group 2 has 30, and group 3 has 30. Spell it out for me in the first one, and then I can see what you're getting at. Group 1, and then G2 and G3. So divide your people into the groups. Once you get your people in the groups, what do? Treatment. Your first treatment would be, pick one of them, low carb, low fat, and then we'll have a control. Low carb. These people get treatment 2, which is low fat. And T3 will be our control. And what are we trying to compare? What do we know about? What do we want to know about them? Yeah, who lost most weight? So we want to compare the weight loss in each of those groups. In your explanation down here, it does tend to get a little bit lengthy, but I need to get you to go through this process and learn how to explain this, because this is a good quiz and kind of test question, supposing we test on this. So how does this random assignment occur? How are we going to randomly assign these 90 individuals to the 3 groups? You could give them all a piece of paper. You could use your random numbers. So whichever way you want to explain that, we can do that. The piece of paper is an easy one to explain. I want to walk through with you how do we explain if we use a table of random digits. So what do? We assign volunteers numbers 0, 1 through 90. So give everybody a number. And then we use the table of random digits to do what? Randomly assign volunteers to each group. And here's what we looked at yesterday. How do we use the table of digits? How do we explain that? So you're going to search for 2-digit numbers between 0, 1, and 90, and you're going to skip the repeats as well as like 0, 0, and numbers over 91. And what I would do is that when I come to my first 30 numbers, I'd put them in group 1. The first 30 numbers, what do I want to say, consist of group 1, the next 30, group 2, and the final 30 will be the control. And really, once you get your first 30, you're like, oh, the rest of you, you don't need to keep going through the tables. Like the rest of you, you're in the control group. So what do we do then? We've talked our way through here. We randomly assign them to their group, which is the treatment. And at the end of the year, because this is a year-long process, Well, what am I going to do with the 30 people in the low-carb group, and the 30 people in the low-fat group, and the 30 people in the control group, in order to compare weight losses? Say what? Look at how much weight they lost, and how do I look at that in this group? Do I look at all 30 individuals? What could we do? We could find the average weight loss in each group, and then kind of compare from that way. At the end of the year, find the average weight loss in each group and compare the results. Kind of lengthy, a lot lengthy, right? But this is a process we have to go through when you're like designing an experiment.